So what we're going to do is we're going to do sort of the first half. We're going to talk some about the modulators part. We're not going to spend as much time on the nonsense and rare because I know there's a whole breakout on that session, and most of you might have already been there once. Then the second half, we're going to do sort of more of the symptomatic therapies. But let's go through. Some of these slides are going to be familiar, but I'm just going to make sure that maybe provide a little bit more detail about them. But then also for each section, we'll just say, hey, is there any questions, comments? Does everybody understand? So we take suggestions, too. We're always open to suggestions. So, I think this slide I showed earlier is just a reminder that everything that we've seen in terms of progress has been great, but we knew that we were building to something, right? And that was this idea of building to the triple combination, which includes really taking that Symdeko combination of tezacaftor and ivacaftor and adding this third drug, 659 or actually it's 445. It looks like it's an old version of this. It's actually 445, not 454. That a couple things here. 659 and 445 are what I call sort of kissing cousins. They're basically built on the same backbone, the same structure, but they've been modified to try to optimize or try to make them work as well as possible. And so that's one of the reasons why it doesn't make a lot of sense to take both of them forward. What they're trying to do is they said, this is the mechanism that works well. They had their chemist make some changes. Sometimes those help affect the way they're absorbed, how long they last in the system, maybe minimize side effects. But they're actually pretty similar at their basis. So I guess I'll just pause here for a second. Does everybody sort of understand this? Any questions, concerns, suggestions? Going once, going twice? Next. So this... The triple combination results, I think a key thing here just to realize is that it's not often that you get to see the results from a trial before the trial is really done, right? Normally what you do is you have a 6-month trial. This is the case with all the other trials before. You wait till the end, you roll out those results. This trial was actually designed differently, and some of this was in conjunction with the FDA to say, let's try to get an early readout after 1 month to help us figure out which of these 3 drug combinations would work best. Because if there's one that's a clear winner, we'll go ahead and make that selection. We'll go in on all that. Well, let's look at those results. This was the 659 and with tezacaftor, ivacaftor. And this is 2 different trials I'm showing here. One is for people that have an F508 del and that stop mutation or something that doesn't make protein, who aren't on anything. And when compared to placebo, the 659 triple combination, people's lung function went up 14% in 28 days. A big drop in sweat chloride, a big improvement in quality of life. The second line is actually a separate trial using, again, 659, but this is people who are already on Symdeko. Now you add in this third drug, they go up 10%, so they'd already had some benefits. But I think the key thing there is 2 different trials, 2 different patient groups, but really just the 28-day data. These are trials that are still ongoing. This is a 6-month trial that we're getting an early peek at. Does that make sense? One of the questions was, if we get an early peek, will we see the winner? The answer is they both worked amazingly well. So this is that exact same setup with the 445 combination. Again, 2 different trials here, 28 days. The first one is this one with F508 del, and compared to placebo, that's a 13.8% improvement. Big quality in life. This other one's a separate trial again. And again, people on Symdeko who now take the 445 version of this, 10% improvement, identical in terms of those 28 days efficacy results. This is one of the things that made Vertex say, hey, we're going to actually keep going a little bit. If there was a clear winner right now, then fine, we would focus on that one. But now what we need to do is people are going to be taking this for years, right? We don't want to actually just base everything on a 28-day time point. So those trials are 6 months, will be wrapping up in May. They're actually already fully enrolled. They're ongoing. But that's really the reason for waiting a little bit for them to make a decision so they can pick the right one. So maybe we can talk a little bit about sort of how they'll do that. But I guess I'll stop right here to see if there are any questions or, again, concerns or suggestions. Laura Gordon.
Yeah, just a random question. Do they start with the same compound? 659 and 445 start as the same basic compound and then just got modified to be...
Yeah. So... Actually, so the question really is, did it start as the same thing and then go out? And so probably there is a backbone or a structure that's they've sort of identified as being the key part of some of the activity. Remember that they actually started out with 4 versions. So the phase 2s tested 4 different versions, and they're all the same thing that had been modified some to try to make it work better. There was 2 of those that worked best, and those are the ones that moved forward, 659 and 445. The other ones have actually been put on the shelf. And that these, so I would say they're really similar. There's probably some key components that are they're almost identical, but and I don't know the details. This is something obviously that Vertex keeps very close to the vest, and they should. But the whole idea was they have some chemists that are really smart, and if they say, oh, we'd like to have this absorb better, we'd like to have this last longer in the system, they can make changes. So same backbone with some changes there to try to optimize. And then at the end of the day, you can have all the theories you want, all the tests in vitro, but you need the clinical data to really say what's it going to do in people. And that's really why this is so exciting, because you're actually seeing that, yes, this is proving to be that effective. So, I guess any other questions or comments? Did I see one? Yes.
What do you mean when you say a 14% improvement? That would be different in somebody with 30% lung function versus 50%
Yeah, so great. This is actually a great question. This is actually, there's a difference between absolute change in FEV1 and relative. That this is absolute change. Absolute change means that if you're at 30, on average, you go to 44. If you're at 50, on average, you go to 64. So this is not a relative change compared to where you are. Does that make sense?
It makes sense, so that's measurable then, if that's the results they're seeing.
That is the results they're seeing. Now, obviously, this is an average, and but we haven't seen results like this even for G551D and Kalydeco. So this is an absolute change. This isn't a compared to where you start from. That's why it's particularly striking. Yes?
Sorry if other people know this, but what are they measuring for quality of life?
Yeah, so we had a similar question. I think it's a great question. One of the things that's jumped out is that people on highly effective modulators, yes, their lung function goes up to FEV1, that's great, but most people report, hey, I feel better. I feel better. And so the quality of life measurements actually has a series of questions. The CFTR stands for CF, quality of life measure, and the R is the respiratory, the lung domain. That there's a questions there that talk about how much energy do you feel like you have? Have you been missing school? Have you been missing work? Ask about other organ systems as well. So it's trying to look at the big overall picture, not just what happens when you blow into a tube. That's a pretty standardized.
I feel like a kid grabbing the back of the shirt to see what's going on, but it's a cystic fibrosis questionnaire revised, and it's a patient-reported outcome. It's a composite patient-reported outcome, and Mike was kind of alluding to this when he said it looks at multiple things. Respiratory is one, your gastric system, how it does as well, and how you function is one, whether you miss school or work or something is another one. I think there are actually 13 domains, and that's what they call the different things, domains. So it's a big, long questionnaire. The one that's used mostly for the label and the rest is the respiratory part. And that's the one you'll see touted the most. And it pretty much is what kind of respiratory symptoms are improving, cough, sputum production, things like that. So in a nutshell, it's a big, complicated instrument. The most common part they use is the respiratory one.
That's perfect. Thanks for calling that. Just one more. Actually, 1 or 2 more. Perfect. It's great. Yeah.
So this is happening over 28 days. Is the prediction that it's going to continue to increase FEV1s, or is that just kind of wait and see what?
Well, I'll take these. I think we'll trade to Tony in a second. So if you look at all of what's happened before, that modulators actually are pretty rapid in effect. That actually oftentimes you see effect a week in terms of lung function. Sometimes in sweat chloride you see it right away. But really what happens is you actually see more stabilization. You don't see continued improvement. I think one of the things that I didn't say this morning, which I almost wish I had, is that there's way more than just improving the lung function out of the box, right? So just not going up 14%, because if you went up 14%, but then you can continue just to decline at the same rate, that buys some time, but it doesn't really change the equation. What's happening and what we're seeing more and more is that it's a combination of jumping up, but then actually the lung function is more stable. And so that's what we've seen with Kalydeco. That's really that highly effective therapy that changes the slope. Now, we need more data. We don't have the data for the triple, but everything suggests that it will be similar to the other things, which are quick initial effect and then some more stabilization, but probably not continued more and more improvement over time. Yes?
Just real quick, how big of a sample or big of a trial do you do something like this?
Yeah, actually, Tony, you remember the exact numbers on that? I should know this. It's about 100.
The two studies were pretty different. The ones in the 508s, and the ones in the 508 with the minimal function mutation. And I'm not doing a very good job in remembering that either.
But it's in the hundreds. It's actually about 150.
The 508 was fairly small compared to the other one.
Pretty tight mean.
So yes, I know. So you're asking about the statistics. Put it this way: this was, you can go onto the CFF website and actually provide a few more details. But this is there was actually a pretty tight mean. As a matter of fact, that bell curve around that was actually something that it's been striking how most people get benefit. It's not like there's one group. There's not two different responder groups, right? There's not one that responds and one that doesn't. That has actually been pretty consistent. The reason for some of the different sizes in this is actually because of the 659 and 445 have such big responses in those with just a single F508 delta placebo. You didn't need that many patients. Remember, Orkambi had 1,000 patients because they were trying to measure a 3 or 4% difference. These results are so big that they feel like, hey, we didn't need that many. This allows us to go faster to get the study done. You still have to build a safety database. I know, Tony, you want to talk about what's required on the safety side?
Yeah, just from what Mike said, in the G551D population, very early on in their dose ranging study, they had pretty much anywhere between 8 and 15 or so patients per arm, and that led to statistical improvements. So the effect was very robust. That being said, there are guidelines about how many patients you should study for how long of a time by the ICH International Council for Harmonization. It's a clinical study kind of thing that's international. And you need data for a year, typically. You also need data in the regulation. It's 100 patients studied for a year minimum. That gets waived frequently in rare diseases like CF because sometimes you can't find 100 patients. CF is a larger rare disease, a less rare disease. So they've always had a fairly robust patient population.
Yeah. So I think just to address your question, sometimes we get trial results and we sort of go, yeah, I think they're positive. This is actually one that there's just no question. And so really now the question is, okay, safety over time, maintenance, no surprises. But the efficacy part, there's just been no question from anybody, even the biggest cynics. All right, maybe we'll keep going here a little bit. And this is when I'm going to ask Tony to weigh in quite a bit because obviously he's used to doing this and being on the other end. We talked a little bit about timelines this morning, but I thought it was just worthwhile one more time just saying, so, right now, as we talked about, they're going to wait for some additional data to be able to decide whether the 445 or the 659 combination is the best one. Those trials are enrolled, and they said they should be able to have that data by May. That will actually just allow. What will they look at? They'll look at efficacy, although efficacy looks pretty similar in some of these. So there may be other things to look at, such as maybe it'll be looking some at sweat chloride. Maybe it'll be how often do people get elevated liver function tests. It's really trying to look for any hints that one's better than the other, and then they'll end up picking that. But that's a big difference between picking that and actually putting together the application for the FDA. We were talking about this a little bit earlier, but Tony, you wanted to talk about, so it's a big lift to actually, it takes a lot of time to be able to get this into the FDA. So you want to talk a little bit about what those applications look like?
Well, there's components of an application that cover the drug's development ever since the first rat that was dosed with it to the largest phase 3 trials. So the files, electronic files, are in multiples of gigabytes. Probably 10, 15 years ago, there were paper files. Everything was paper. And it would be 2 library shelves filled with like 200 volumes of giant things that were like 300 or 400 pages apiece that if you were going to review them, you had a table of contents, and then you just wandered through and grabbed volume after volume to take a look at. So it's a lot of information, not just on the clinical study, but how the drug is made, how many rats died, the whole thing. So
I just highlight that because obviously they're doing a lot of that work right now and getting it ready, but some of that the explanation for why they can't just the next day turn that in. But that'll mean that probably the third quarter is what Vertex has been talking about for submitting that, and then that would start the clock for the FDA review. Dr. Hodge.
Yeah, I have a question on the submission, which is, it's been put in with a specific set of mutations, right?
Well, that's open to discussion. Well, so keep going. Keep going with the question, yeah.
So the question is, why are they doing it that way, rather than just say, if you've got one del 508, doesn't matter what's on the other side. But my understanding is that's not how it's going in.
Well, so I think that's a little premature, truthfully, to make that statement. Yeah, so the question is, when the submission comes, what would the label, what are they asking for, right? Who would benefit from this right away? And so one of the standard things to do is just to take what the entry criteria for the trial are. So you said, oh, this is the group for the trial in general by mutations, that's the group that's going to benefit. Peter's highlighting that it's a little bit complicated here because really what's shown from these test results is that if you have one F508 del, it probably doesn't matter what the other one is, that you're going to likely benefit. I think there's two groups that are going to be sort of confusing and they're going to have to work through. One of those, actually I'll answer your question in a second, but I'm going to start with the ones of people who are already taking Kalydeco. This was in this caveat slide, but people who are already taking Kalydeco because one is F508 del, the other one is something like G551D or something that allows them to take that. They are going to have to we haven't really studied that group yet. We're going to have to demonstrate that the triple is actually much better than just taking Kalydeco alone. I don't know, Tony, you want to comment about that?
Well, just to jump to the chase a little bit, the phase 3 trials and the ultimate labeling are predicated on the previous way the drug was developed, or the drugs. And the populations that were chosen, initially G551D was the most likely population to respond to a potentiator alone. Then added on was what we call correctors, for lack of a better term, and included the 508 mutations. Residual function mutations became a definition. So the program has been rather definition specific. So now we all know, if the data hold on, that the drug would likely be effective for anybody with a 508, because that's all you need with a minimal function. It doesn't make any protein at all. But that being said, you've now got these other pieces that you got laying out there. And one of the pieces is you've got a rule/regulation that says if you have a combination drug that's approved for something and you want to make an addition to the combination, or any drug, in addition to it, then you have to show that what you added gives you some extra bang for your buck. Otherwise, why would you add a third drug when it didn't help anybody anymore? So what they're going to have to do is clean up the indication over time, and what Mike was going to say, I think, is that they've got a study planned to look at G551D and other gating mutations and residual function mutations, and see whether the triple combination works any better than the Symdeko or Ivacaftor or Kalydeco that they're on. So that's the evolution of the program, and that's where it got to where it's going to be right now.
So Peter, I'm thinking about, I just know some of the details about your daughters, and I think they will benefit from the triple. We want to go as fast as possible. They need data to be able to show that it's going to be above Kalydeco. I think it can go faster because all the safety parts, all those things would have already been, the groundwork have been laid, but that trial is going to be ongoing. The flip part of your question, though, is about those people that have one F508 del and maybe minimal function. There's this list of 190 mutations, and you sort of say, am I on this or not? I think there's a lot of discussion about the best approach on that. I don't think that it's, that discussion's ongoing. That's going to be both with the company as well as with the regulators. It would be great if it was as easy as saying one F508 del and anything else. I think someday we'll be at that. We just want to try to get there as soon as possible. Because I think you're right, that's the destination. The question is, how can we get there fastest? Okay. So I guess, and just in terms of the approval part, it's always dangerous when you start putting dates, especially when you're not in control, but there are certain things we know that are FDA guidelines that they have to follow. And so if Vertex submits their NDA, which means new drug application, in the third quarter, that means that it's likely that approval would happen more in 2020. Tony, I thought you did a good job this morning of talking a little bit about the guidelines as well as some of the history here. You want to talk a little bit more about that?
The moment that a new drug application or NDA is submitted, a time clock opens up. And depending on what kind of drug it is and what the population being studied and the indication is, that can vary. For instance, a regular time clock for a drug that's been given to humans for some other disease maybe and is being used for CF or asthma or something like that would have a 10-month review clock. For a drug that has what they call an NME in it, new molecular entity, which means it hasn't been approved for people before, that time clock is a little longer because they think this is something that's never been exposed to people for a long period of time before they want to look at it really closely.
Jump in here real quick, just real quick. I'm getting a message that says my battery's low and about to turn off. Is that a problem? Or is that, as long as we have it up here, it's good. I just want to make sure that it's not going to... Try, keep going. So okay. I just didn't want to suddenly lose our...
So that being said, 12-month clock for a new molecular entity. There's a little caveat here in that if it's a drug that's going to meet a really great need in the population, they can call it a priority review. And almost all CF drugs, especially the modulators, have gotten that kind of review. So that drops the 12-month clock down to 8 months. And then, of course, if the group if the data are incredibly strong and there's no issues at all on anything, manufacturing or the studies or the designs or the inspections of the facilities, then they can even do it faster. And I think Kalydeco was done probably in about 4 months or so. So the guaranteed time, in my opinion, would be about 8 months from the time it's submitted, but it could go faster.
Yeah. So you can do the math. Our hope is it would go faster. So actually I want to introduce Erin Tallarico, who's actually being Carol Merrill, but she's much, much more than Carol Merrill. So she's actually was one of our adult CF nurses for many years at Hopkins and then joined the foundation and actually is helping to lead the lung transplant effort. So we're really grateful for her. So thank you, Erin.
Hi there. So just real quick, piggybacking on what Dr. Hodge had questioned about. So two of my girls with Delta 508 R117H. So originally started Kalydeco a few years ago, but within a few months, their clinic, we switched from one clinic to a different clinic, and the new clinic said, actually, we're not putting them on it because we're waiting to see if they eventually would benefit from something like this that was in the pipeline. So they came off of it within 3 months and haven't been on it since. Would that change anything since they're not currently taking Kalydeco, knowing that my older one is going to be 12, so she would benefit next year possibly from it?
So it wouldn't, because really the guideline, they're in the group that's going to be studied, right? So if they're eligible for it, and that's sort of whether or not they're on it or not is probably a different discussion, but it's really this whole point of we know that they could be benefiting, at least some of it with R117H with Kalydeco. So there's going to be a separate study that says, will the triple be on top of that? So that's probably, that trial's going to be done. It's probably another year, realistically, before that question gets answered. Okay? I'm sorry. Okay. So I think we're maybe got 5 more minutes on this part. I think we've talked about a couple of these key caveats. I think one of the things is we always say we want to make sure that this is continuing to work, right? Again, this is actually the 28-day data from a 6-month study. We're really going fast. As a matter of fact, I think we get a little spoiled in this community because we go so fast. As a matter of fact, when I talk to other people in other diseases, a lot of times they're like, you're doing what? Another drug? You're kidding me. But we have to make sure that efficacy and safety are there, that people are going to be trying to move fast and move it down to kids. One of the things I think that's important to realize is it's not just as easy as just sort of saying, just reduce the dose down and just give it, smaller amounts, that there's actually separate things that you have to pay attention to as you get younger. People's livers work differently. You clear medicines differently. And so that's the reason why it's not just this, let's give it to the kids and just reduce the dose. There can be some surprises. And so, but it will go faster than it has in the past. And I think these other ones we've actually talked to other than this compassionate use question. So the guidelines on that are really, once Vertex selects their drug, they're a 3-drug combination. They've made a commitment to actually ask the FDA for permission to provide compassionate use. That's a 28-day, correct me if I'm wrong, 28-day or 30-day, whatever window the FDA has to get back. They almost always say that's okay. That doesn't mean the next day the people can get drug. What happens is everybody's local doctor then needs to get permission from their local IRB, their local hospital, to be able to provide this. And that takes a little bit of time as well. So that whole process is probably a 2 or 3 month process. And remember, compassionate use really is most likely going to be, we don't know the exact things that Vertex will say, but it's going to be people whose lung function is more severe, less than 40%, people who are on a lung transplant or have had really life-threatening complications where a couple of weeks or months makes a difference. But that's why we say that probably sort of end of summer, fall, there may be an opportunity for people with severe disease to get that early access, assuming everything goes according to schedule. So I guess any comments, questions on that? Okay. Oh, Pam, yes.
Curiosity. You said the doctor would have to apply to the hospital, but I remember when the other things. Oh, thank you. I remember. I'm asking if who actually would you suggest that a patient or the parent go to them? Because I feel like when Orkambi and Symdeko, we have a great care center, but they don't necessarily know the top, like the exact information about the stuff. So
we're gonna be trying to help with that. It's actually as soon as this compassionate use program starts to roll out, we'll actually help to communicate some of that. But really, what happens is you talk to your doctor and you say, "Hey, is there a chance of my getting early access?" They'll have some guidelines, and what they do is they would actually get permission from their local IRB. But at the end of the day, it's an application to Vertex that says, "Can you provide this drug? Here's the thing." If it's a legitimate application, they would say yes, but there is that extra permission step that it's not just quite as easy as sort of asking Vertex for it. Yes, but advocate. Thank you, Pam. That's just really the question is, not everybody automatically says, "Oh, we could look into this." But if it's one where weeks makes a difference, months make a difference, absolutely, if it's eligible. So let's maybe we'll bring this home because we're going to move to the other part. I'm going to actually skip through this because we've talked a whole bunch about this. Just mention that there are other companies, and we're really trying to help them be successful in this space. Proteostasis announced some results. They still have some work to do. AbbVie, I list these two because these are ones that are both in clinical trials. But we know there's some drugs out there that are actually better than what are in patients right now. We just actually need to help them get forward. And so maybe what we'll skip now to is this idea of treating complications. And part of this is because we know that even with these incredibly exciting modulators, they're not going to solve everything, right? People are still going to have scarring on their lungs. They're still going to have other problems. So, Tony, you want to come up and sort of talk to us a little bit about that? And we'll do sort of the same format. If you have questions as you go through, let's make this interactive. And at the end, we'll throw it open for whatever questions people have.
Hi, Hannah. And I know that, technically we're supposed to finish up at a certain time, but, I'm fine with hanging around for as long as anybody needs to. If they need to ask a question, I think Mike probably feels the same way. So don't feel like you have to jump in and get your question answered right away. If you're missed, it's a problem. So I think this slide was, supposed to get shown this morning, but it was one of those things where I think it didn't quite click well enough. And it's one of the more important slides I think that you can take a look at and digest a little bit. So it shows how we think the effect of CFTR modulators are going to have on the CF population over time, over the next 20 years or so. And I think it's important to look at the bottom first, and then we'll work our way up. Look at the dark blue, and it's incredible that you're going to see a gradual increase in the population of patients, people with CF who are healthy, completely healthy, normal lung function. Then there's going to be a population, light blue, and a population that is in the yellow phase here, that are people that fit in a couple different categories. Some people already had significant lung damage, and taking a modulator or something like that is not going to make your bronchiectasis go away. It's not going to reverse lung destruction. It's going to hopefully hold it in its tracks, and you're going to go forward, but you're still going to have something and some chronic inflammation. I think we've kind of said, you might have something like non-CF bronchiectasis, which is a disease entity that people with damaged lungs for various reasons have that need chronic anti-inflammatories and other drugs as well. And then there are patients who are going to have severe lung disease and potentially need a lung transplant. So the people that have lung disease that already exists, and the population of CF patients that really aren't eligible for a modulator or any other CFTR augmented therapy right now, are the ones that we're really going to need to develop these anti-inflammatory and anti-infective medicines for over the next 5, 10, 20, 30 years. So I'm going to be known as the pipeline guy by the end of this the talk, but I'm not going to get into this. This just shows you the breadth of the CF pipeline. And Mike talked about the modulators on the left side. All the other ones I'm going to touch base with really quickly in the next 10 or 15 minutes. It's on the CF website. Click the research tab at the top, go down, and it'll say drug pipeline, and it's pretty much this exact same thing. People already know, I think, about the CF Infection Research Initiative that was announced at the NACFC last October. We've committed over $100 million over 5 years to help improve detection, diagnosis, treatment, and outcomes of people with infections who have CF. To put it in the context a little bit, this is about twice as much as we had done previously. So this is about a doubling of the funding that we've been doing. I'm going to go through this. What do we call these things again? Pipelines? Yeah. We're going to go through this pipeline kind of quickly, then I'll stop and see if anybody has any questions on this first part. Our anti-infective portfolio right now crosses across all phases of development, from discovery through phase 3. All the way on the far side on the phase 3, Savara is finishing up a study, hopefully soon, because it's been taking a while, on inhaled vancomycin to treat methicillin-resistant Staph aureus, which can be an organism that causes problems with people with CF. There's some other really cool things going on in the anti-infective portfolio. The ones that are in purple are drug therapies that have the potential to kill multiple I'm going to call them germs. I told the earlier group, I like germs better than bacteria. It kind of makes. But the issue with those is that it's actually bacteria germ agnostic. So one is inhaled nitric oxide. Inhaled nitric oxide is already approved for the treatment of primary pulmonary hypertension of the newborn because it's a very potent vasodilator. That's the research I did for 15 years before I segued out of it because it wasn't good enough to do it. But that in higher doses is an anti-infective and anti-inflammatory agent. So it's being studied right now by Novoteris in phase 2 trials. The other one that's kind of cool is a contrast agent for MRIs called gallium. And gallium looks a lot like iron when you look at the table of periodic elements. And iron is an essential metal for bacteria to survive and grow. It must be a cofactor in an enzyme system or something like that. But gallium is picked up by the bacteria thinking it's iron, but doesn't have the same activity. So basically, the bacteria can't metabolize or can't continue its growth, metabolism, et cetera. It dies because gallium is not working like iron would. So that is interesting. It's in IV form, and it's also being developed in an inhaled form. So that's really cool. The other one I got to bring up because I think it's kind of interesting and kind of funny in a way is, I only thought that bismuth was in Pepto-Bismol. But they got one here under the preclinical microbiome that is a bismuth thiol that apparently has anti-infective properties. But I will wonder. Anybody who's taken Pepto-Bismol, at least it has a tongue like mine, it kind of turns black. The one other thing I'm going to mention, because I think this is one of the cooler ones in the portfolio also, is by Sound Pharma. And there are people with CF, especially older people, that have a lot of hearing problems because of the chronic, over time use of IV aminoglycoside antibiotics. And this is a drug that could help prevent that ototoxicity. It would be given at the time somebody was going to get a course of IV therapy that included amikacin or gentamicin or tobramycin, and it would, I think this is the way it works, it would block the pores in the hair cells in your cochlea, and the aminoglycoside wouldn't have access to the inside and would prevent ototoxicity that way. So that's something that's just started being dosed in people with CF right now. One other thing that's kind of important right now in the CF community and getting more important is the issue of non-tubercular mycobacteria. And that is not just a CF thing, but people with immunodeficiencies have issues with it. People with other kind of lung diseases like COPD have issues with it. So we have set up a consortium of about, I think it's nine rocket scientists around the country who their goal is to study non-tubercular mycobacteria and how to best diagnose and treat it. And that's a big deal. The issue with it is that, like TB, you have to treat it for a very long time. And the problem is that the drugs that you need to treat it with for a very long time tend to be very toxic. So you want to make sure, since this type of disease process is very slow and indolent in a lot of people, that the treatment is not worse than having the germ in the first place. So that's some of the stuff that's going on with regard to that, to try to figure out when to treat and when not to treat. And there's two types of it. This MAC is called Mycobacterial avium complex, and the other is called Mycobacterium abscessus. And they're the two most common ones that you see in the U.S. Helping along the way are multiple companies. And the cool thing is, you've got companies like Savara and Aridus and Novoteris, which are very small companies that are doing trials, some of which I had mentioned. But you also got some big guys in there, like the Gates Foundation is interested in this. And Johnson & Johnson, which is the biggest drug company in the world, is interested in therapies for this, which gives it a lot more oomph than just a couple biotech companies running out of a garage. So I'm going to pause right here, and if there's any questions, we can go here, or otherwise I'll move on, but move forward.
Thank you. It's probably not a question specifically to this, but I'm curious about any research being done on gene modifiers and how that affects the research you're doing with the medication, and or how it's affecting cystic fibrosis even prior to the medications fully being given to patients.
I'm going to have to pass on that one. You got any thoughts?
Yeah, I guess a little bit.
Who's got thoughts?
No. So I think you asked about genetic modifiers. Genetic modifiers are other genes besides cystic fibrosis gene which can influence how somebody's doing, and they're probably into two categories. One is those which might influence just how severe CF is, but then there's other ones that might influence susceptibility to infection. And so I think there's actually been a lot of work in this area. Mike Knowles at North Carolina has actually had a big project trying to look at other modifiers which influence severity of lung disease. There's been a couple ones that have been identified. Another one, there's been a lot of research about mannose-binding lectin. It's another gene which can affect your immune system. But the truth is we haven't been able to find any other smoking gun that says, oh, this gene really influences. It's probably a lot of different genes in small amounts. So a good amount of work, but probably nothing that we really say, hey, we need to go after this.
The slide that was up just before this one, the anti-infected portfolio, I saw that actually this morning, and it was very interesting, I think, to all of us because it's so comprehensive. It's not just one area of research. I was looking on the CFF.org website, and I didn't see it. Is it up? And if it's not up, could you guys put it up? Is that something that might be possible?
I think it's possible. It's obviously public information because we wouldn't put anything out in public like here if it wasn't. So sometimes we make these things called infographics. I just learned it since I've been at CFF. I didn't even know what they were, but we can talk and think about that. There's another thing that we have is that we've got some. I guess it's a set of infographics that we've used at different family days, that could be distributed that we would update every year. So that might be a possibility also. But we can either get back to you or with that kind of information, and we'll try to figure it out.
You can also dig into some of that. Tony showed that one slide that's actually the massive pipeline. They're listed in a different way within that. It's not quite as clear, but you're right, maybe there's a better way of presenting it, as Tony said.
We're going to pivot slightly and move on to the anti-inflammatory products that we're looking at right now. And again, they cross preclinical to phase 3. The one in phase 3, it's not quite in phase 3, is a company called Celtaxis. They're discussing what a phase 3 trial might want to look like in this kind of population. There's not as many anti-inflammatory agents in the portfolio, but they tend to be more high-risk programs as well. They're not as well defined as I'm going to eradicate Pseudomonas. That's a very targeted therapy. If you look at what a inflammatory cascade looks like after you get cut or pinched or have a bacteria in you, it's just a stimulus, and then things go like this, out like a giant wave or ripple effect. So there's probably hundreds of anti-inflammatory cytokines and other agents that modulate that response, and that's why it's such a hard problem to pick the one or two that might turn the whole thing around. But the acebeluzstat works on the leukotriene pathway. There's a couple asthma drugs named Xylutin and Singulair that work on that same pathway. So it's a pathway that's been used before for, allergic and inflammatory diseases like asthma. Corbus is in the middle of a big phase 2 trial, which hopefully will find a dose for them. And linabazum is a cannabinoid. There's a lot of buzz around cannabinoids right now. Cannabinoid oil, CBD, and this, just to make things clear, because the company gets really upset if you think it, and you call it a not cannabinoid, but if you think it has some side effects like marijuana would have, it doesn't. There's no sedation, there's no highness or anything like that. It's a cannabinoid agonist, which has been shown to work, and it's in vitro and is in development for about 3 or 4 other diseases. So there's people that are buying into it. Laurent is developing fenretinide. It's a vitamin A derivative, and it also works on the arachidonic acid kind of pathway schema. That's on the leukotriene inhibition is in that umbrella also. But what the interesting thing is that it's probably more closely to what an ibuprofen type drug would do. And we've got some precedent with ibuprofen. Santhera is studying a human neutrophil elastase inhibitor. So you've got neutrophils, which are white blood cells, which accumulate where infection is. And part of the way to get rid of infection is to spew out enzymes and things like that that would be antimicrobial in a sense. One of those is neutrophil elastase, which is an enzyme which pretty much dissolves a lot of stuff. And one of them is elastin, which is lung tissue, which is not good for your lungs. And this kind of tamps down that inflammatory response from that. It hopefully preserve more lung tissue. And Parantha is really kind of still in the garage, and they're looking at TGF-beta, which is a kind of cytokine superfamily that might have anti-inflammatory and anti-fibrotic potential. So that's the anti-inflammatory portfolio. Any particular questions on any of them, or any other thoughts? Otherwise, I'll kind of just move through and we can cover things. We also do some mucociliary things. Right now there's hypertonic saline, and it's a long, arduous kind of process, and a lot of times it doesn't taste very well and makes you cough. There's an inhaled mannitol, which is a powder, and it's called Bronchitol. It's approved in several countries. It's approved in Europe. I think it's also approved in Canada, and it didn't get approved in America probably 5 or 6 years ago because there was an issue of whether it was safe in certain populations. There was an increased episodes of hemoptysis when people took it because they were coughing so much. and some way the study was done, whether they could draw conclusions from it. But hopefully we're thinking that they come back again and visit after they've gotten some data from Europe and Canada and can, I guess, respond to questions that were brought up about that. Oligo-G is made from seaweed, and it is like a calcium chelating agent. It binds calcium, and apparently when you bind calcium, it makes the sputum get thinner, and therefore you could expectorate it better. I don't know if I would call it a super guaifenesin, Mucidex kind of thing or not, but we'll have to see how that goes. The other ones are called ENaC inhibitors. Sodium channels actually pump ions the opposite way of the chloride channel should. And by inhibiting them, you can prevent that. You can prevent some airway dehydration and hopefully keep the airway more liquid, so you can move mucus out better. So that's the mucociliary clearance aspect of things. Yes.
How do these compare to clomazine, standard mucociliary clearance? And are any of them dry powder?
Let's see. We know the mannitol is dry powder. Oligo-G, I think Oligo-G is, right? Yeah, Oligo-G is. The other ones, I don't think they are. I don't think anybody's ever looked at them compared to pulmozyme. Pulmozyme works because DNA is thick and tenacious, and it kind of cuts up the DNA into smaller pieces and makes your sputum, which has a lot of neutrophils DNA in it, thinner. But I don't think there's any aspect that one would potentially replace it, or is it any better than it right now? Again, some of these things, the only one we have really decent data on is the inhaled mannitol. All the other stuff is very early data, mostly related to safety.
And that studied a lot of those patients who were on pulmozyme, so this was sort of in addition to.
Just real quickly, there's some other multi-center trials that are being done or are going to be started soon. You've heard a little bit about the one called Galaxy, and I think that it has to do with the assessment of GI symptoms in a study. And a couple years ago, when there was a big question asked to the CF community about what kind of things are you most concerned about, what bothers you the most, GI symptoms were way up to the top. Lung symptoms were up there, but GI symptoms were above that. So this is a way to try to answer what GI symptoms need to be assessed as endpoints in studies, and what can they be looked at when you have an intervention. This is a study that hopefully will help answer some of those questions and see what kind of GI symptoms are important and what can be measured in CF patients. Check CF is a really quick study. It's important scientifically, though. People get their sweat chloride checked. Pretty easy thing, I'm sure most of you probably know. And then they go on their modulator, and they get their sweat checked periodically thereafter. And the key part about it is that over time you can try to see how much of a change in sweat chloride when they started that modulator, how does that correlate with, or is that associated with their clinical course? And that's really important, because sweat chloride is a quick endpoint in a clinical trial. You can do a sweat chloride after 2 or 3 days of a modulator, or a week or so, whereas even FEV1, you need to do it for about a month or so to make sure you've got it. So it's a really good study scientifically, and it's actually quite easy. Glutathione is an antioxidant, and we talked a little bit about it last time about whether glutathione is better than blueberries or anything like that as far as getting your antioxidants for the day. But there's some question about whether supplement of antioxidants can help, especially children, with their growth and weight gain. So that study is ongoing. I think it's out of the University of Colorado mostly. And then there's, of course, the STOP2 trial, which is to assess how long exacerbations need to be treated with IV antibiotics. Can you get away with 1 week? Do you need 2 weeks? Do you need longer? Which is really important, again, from that ototoxicity, as well as missing school or work or all those other kind of things that would happen when you're at home sick or in the hospital sick. So I think that's pretty much it from this quick blaze through the non-modulator space. So we've got some time, I think, don't we?
We do, and actually, I'm going to start off by maybe hitting this one question that comes from online.
Okay.
And then we'll just throw it open. So anything's fair, anything that you were wanting to ask. So how does the triple impact the GI tract? Are enzymes still needed with every meal?
That's a good question. You know, I know that the modulators that are approved increase GI tract bicarbonate, which is really important in you know, how enzymes work, because acid breaks down the enzymes, and that's why you probably have to take so many of them, because there's not a good way to protect that enzyme from getting broken down at low pH. So that's one of the primary things, and that does help. And obviously, the aspect of people on highly effective modulators like Kalydeco, when you gain 2 or 2.5 kilos, it's doing something pretty good to your GI tract. And probably it's related a lot to the bicarb thing.
And the only thing I would add is that in the question about taking enzymes, the answer is if you're an adult and you're taking enzymes, you still need to take your enzymes. Things may work better, as Tony was saying. But this gets back to this whole vision. Sorry, maybe I'll stand up because I'm saying, no, we both stand up. This vision, yeah, exactly.
We're on the camera.
There we go, it's great. This vision of preventing problems. And so there are data now, and you may have heard about this, but that if you're able to start highly effective modulators early, really early, as in age 1 or 2, that you may actually be able to prevent the problems that happen in the pancreas. And so I think in the future, there's a possibility that modulators could change this by treating people before they develop the pancreas, and they may not need to take as many, or even maybe not at all, enzymes. We have to figure that out. But the answer is, if you're on enzymes now, the modulators probably aren't going to change that. It may just make things work better.
Tell me about the ferret paper.
You want to talk about the ferret? Okay. Well, so we were just, literally this week, we were comparing notes, throwing around. There was actually a recent animal study in ferrets. So there's a ferret model of cystic fibrosis, believe it or not. That's actually a very good model. It comes out of Iowa. And that's ferrets everywhere in Iowa. No, just kidding. It's that's right. But the bottom line is this ferret model, what they did was actually to treat the ferrets in utero before they were born with Kalydeco. So these are ferrets that have G551D, cystic fibrosis, treated with Kalydeco in utero, and they never really developed cystic fibrosis at all. Their pancreas was normal, fertility was normal. Basically, they didn't have problems. So it just sort of hints at why we're thinking early treatment is such a big deal. We're going to learn a lot. That's why it could really change some things. So questions, throw it out there. Let's just actually...
a comment and then a question.
Okay,
so
and then we'll make sure we'll get somebody else as well.
Okay,
Perfect,
and based off of what you just said, my daughter was diagnosed at 4.5 years of age. She wasn't caught. And the first 2 years of her life, I could keep the weight on her. And it was after that we started having the troubles, and we almost lost her because things got bad. But so that just proves, I think, exactly what you're getting at, that if caught earlier on and treatment starts earlier on, you can maybe prevent things. My question's on the glutathione. Andrea, my daughter, was diagnosed in 2001, and glutathione was being talked about back then. Yep. But nothing that I read or saw within the foundation was being tested. So the question is, how long does it take for something to start getting tested, even if it's been talked about anecdotally through other populations that aren't even CF related, but somehow got into the CF population? And why does it take so long, maybe, for the foundation?
Maybe I'll take this one because I know the history here.
Okay.
So when it was originally being talked about back in the day and everybody was buying it at the health food store, it was talking about it's inhaled medicine. It was talking about it's an inhaled medicine. This is not a trial. We've actually done a trial where we've looked at the effects of inhaled glutathione, and truthfully, it doesn't work. This is a different study. So that we actually pretty quickly, the foundation sort of put together and tried to say, is this really working? Because you're right, it was hot. It was on all the chat rooms, right? People were sort of saying, it was sort of the the flavor of the day in terms of wonder drug. But that actually, unfortunately, is not panned out. We'd love it, it will, but it didn't really make a difference in the lungs. There was some observations, though, some people were kids, as Tony was mentioning, that they were actually taking it orally, and that it seemed to actually help with inflammation in the gut and help with weight gain. So the trial that Tony's talking about is actually not, it's a little different variation than what was originally described. This is actually one that would help with nutrition. And we should actually know in this next year about this. So it was actually, it's a follow-up to the original follow-up of that, but it's a different flavor. So.
We have 33-year-old twin sons, and we're raising money for lung function. So don't get me wrong, I completely understand the importance of that. But in addition to that, I work in primary care, and I also see the devastation of diabetes on a daily basis. And our kids were diagnosed with CF-related diabetes 20 years ago. But when I compare their insulin needs to a type 1 diabetic's insulin needs, it's much, much less. So what is the prediction of the potential for retained beta cell life and how modulators may help eventually get some of these patients off of their need for insulin?
Yeah. Well, I can try to help you out with that a little bit. The pathology of CF is mostly with epithelial lined tubes, right? And that is mostly the exocrine pancreas stuff. That's why you need enzymes potentially from a very early age. The beta cells are outside of that system. They're in the pancreas, but they're in their little islets inside the pancreas. And I think it takes a long time for those islets to fibrose because of what's going on in the pancreas, and gradually get to the point where you need insulin. So I guess the hope is that if you can improve function of the exocrine or reduce the amount of impact that exocrine pancreatic dysfunction has on the rest of the pancreas, that you might help prevent either CF-related diabetes, or at least, go 5, 10, 15 years longer before you get it. I think that's the way I think about how that might work.
Yes.
Probably not.
Yeah, I was saying on the flip side, unfortunately, there might be agreed, probably not, is really what it comes down to. That we've actually done one study, is we've supported a study out of Penn that looked at whether or not these modulators will actually change the ability to secrete insulin, all those type of things. It helps a little bit, but it's almost a timing thing. As you were saying, is once you get too far along, you can't undo it. But if you could start early, you might be able to prevent it. So I think most likely what would happen is with these highly effective ones, it might reduce some of the requirements. It might make things a little less brittle, but the likelihood of actually going from being on insulin to off is pretty unlikely.
Oh, sorry.
Hi. Excuse me. My wife can no longer take Advil, I believe because of her kidneys. And right now for pain, she can pretty much only take narcotics or something along those lines. I was curious if anything in the anti-inflammatory pipeline might potentially be an option for her.
I don't think any of those anti-inflammatories are going to be analgesics, to be honest with you.
Yeah. So I know your wife very well. Tell her hello. I think there's going to have to be other approaches. I think the anti-inflammatories are going to help with other things, but I'm just not picturing the pain part. Even the cannabinoids, as Tony was highlighting, are not ones that actually affect sort of pain receptors or mental part, and it's actually one of the advantages. So maybe we can talk afterwards and sort of compare notes to some other things to be able to help Rebecca with.
Along the lines of the diabetes, does the triples help with CF-related liver disease at all?
I don't think anybody's going to know for a while. I think that we're developing new tools to assess liver disease, because right now it's kind of like, the disease I used to do a lot of research in, pulmonary hypertension. If you're an adult with pulmonary hypertension, you don't know you've got it until you're very, very, very late stage. And that's kind of the way I think about the liver disease. You have a variceal bleed and you go, oh, we have a problem here. But there's some new techniques to try to get a really early sense of any kind of people that are at risk even for liver disease that we can kind of use to see if a modulator might be beneficial in that sense. I would hope that it would. I mean, just a general sense of how it would work and what it would do, but I don't think anybody has the real data on that yet.
But we're going to study. That's part of the PROMISE study. And agree that it's something that makes a lot of sense that it could be helpful. We just don't overpromise until we see. It also depends on what stage it's in. So if I take one online, and then maybe then it's time to actually probably. I think Tony and I are both happy to continue to answer questions after here, and come on up. But I think that we need to wrap up a little bit. So this one says, if somebody wants to get into the 6 to 12 age trials for the triple, what's the first step? So what I would say is go to CFF.org clinical/find. And there's a clinical trial finder, which actually you can sign up for and get updates whenever new studies open. I always say talk to your own doctor, talk to your own local center. But if you sign up and actually you can make requests that anytime a new trial is being started, that you'll actually get notified. And you can wait to see when the 6 to 12 study actually gets announced. And then they'll actually have a list there of all the different sites that are doing it. Obviously, you want to most ideally focus on your own site, but it'll list all of those. You can actually even click on it and express your interest. There's an I'm interested button that sends an email to that research coordinator in that study that says, I'm interested in this. That doesn't enroll you in the study, obviously. It just sort of says, please contact me. But that's probably a good way to start for the person online who asked about that. So that's a great question, maybe a good one to wrap up on. So, yeah. Tony, you want to wrap us up?
Just to that point a little bit. In my mind, if you are really interested in that and want to get a first jump on it, I would start paying attention and talking to people as soon as Vertex picks the drug they want to go forward with, because they're not going to study in pediatrics a drug that is not going to have the benefit of ever being approved for that child. It's kind of an unethical kind of thing. So as soon as they make the choice, if I was interested in that, in getting somebody enrolled in that, that's when I would start to move.
Start to look. Well, so guys, thank you. It's been a wonderful 2 days.